Students, now let us see the last step of aerobic respiration, that is electron transport system and oxidative phosphorylation. Okay? And in the previous three steps, what we followed? And in the first step, glycolysis, one glucose split into two molecules of pyruvic acid and the energy liberated is packed in the form of NADH2. And whereas the second step, that is conversion of two molecules of pyruvic acid into two molecules of acetyl coenzyme A that occur in mitochondrial matrix also yielded two molecules of NADH2 we followed. Now and recently and we discussed about that how that two acetyl coenzyme A undergo complete breakdown there in mitochondrial matrix through Krebs cycle reactions, and there in Krebs cycle reactions we observed for one glucose 6 NADH2 molecules, 2 FADH2 molecules and 2 GTP, of course later may converted into ATP and available to cell. So, I mentioned ATP here, 2 GTP by SLP are formed. So, as we summarize the total aerobic respiratory pathway, by the end of Krebs cycle reactions we observed one glucose molecule finally and completely broken down into carbon dioxide and water and been completely oxidized, and we followed 6 oxidation steps, one during glycolysis, one during link reaction and 4 oxidation steps during glycolysis. So, glucose by 6 oxidations fully broken down into carbon dioxide and water. The main theme of aerobic respiration is completed actually by the end of Krebs cycle reactions. But what is the significance of the fourth step, and that is electron transport system and oxidative phosphorylation? Let us discuss. As we verified, the energy liberated in various oxidation steps as well as in SLP reactions is packed in the form of NADH2, FADH2 and of course ATP. For one glucose, as we calculated totally by the complete breakdown by the end of Krebs cycle, 10 NADH2 molecules are added to matrix, 2 FADH2 molecules are added to matrix and 6 ATP are formed by SLP. During that 6 ATP, 4 ATP formed during glycolysis, whereas 2 ATP, that is in the form of GTP, added to matrix formed during Krebs cycle. But as we verify, is the essence of aerobic respiration or the main motto of aerobic respiration is completed by the end of Krebs cycle reactions? No. Because the main motto of aerobic respiration is to convert the energy in the form of glucose into available form of chemical energy, that is ATP. But here as glucose undergo complete breakdown and by the end of Krebs cycle and only a small amount of energy is packed in the form of ATP and directly available to cellular activities. As major part of energy of glucose is packed in the form of NADH2 and FADH2. As they are added to cell and are they serve as energy currency and can be utilized for cellular activities? No. So, NADH2 and FADH2 are not readily available form of energies. So, the energy packed in the form of NADH2 and FADH2 again should be released and should be utilized for the synthesis of ATP, mean the packed energy again should be transformed into ATP, then only available to cellular activities. So, to save that purpose, and here oxidation of NADH2 and FADH2 occur, and that is electron transport system, and due to oxidation of NADH2 and FADH2 the electrons and protons liberated and they are received by the oxygen, and oxygen you know play a great role in aerobic respiration, and only in presence of oxygen link reaction and Krebs cycle takes place, does not occur in the absence of oxygen, but oxygen do not play any direct role there in link reaction and Krebs cycle we follow. And the direct role of oxygen is in electron transport system. The electrons and protons liberated and they are received by oxygen here, and that oxygen is reduced to water molecule. But during this electron transport and proton translocation happens according to Peter Mischel chemiasmatic theory that causes the synthesis of ATP through coupling factor, that is F0, F1 particle. So the total process as we represented here, of course in brief let us follow, and that is oxidation of NADH2 and FADH2 liberate electrons, and that electrons they are carried through various electron carriers and cause proton translocation. That proton translocation results the synthesis of ATP. So the energy liberated by the oxidation of NADH2 FADH2 is utilized for the synthesis of ATP, and so that kind of ATP synthesis which is favored by the oxidation of NADH2 and FADH2 is known as oxidative phosphorylation by the involvement of coupling factor. So due to oxidative phosphorylation, that energy is again packed in the form of ATP and readily available for cellular activities. Okay? Students, now let us see what happens during the oxidation of NADH2. How that energy packed in the form of NADH2 is utilized for the synthesis of ATP, and what happens if oxidation of one NADH2 takes place and cause the synthesis of how many ATP. And here we follow the mechanism of electron transport system for the oxidation of NADH2 I represented, and actually electron transport system and oxidative phosphorylation occur in which part of mitochondria? Let us see first, and that occur in the inner membrane of mitochondria, and you know infoldings of inner membrane of mitochondria are called cristae. As we recollect the diagram of mitochondria, all of you see here, and this is outer membrane of mitochondria. The inner membrane of mitochondria show infoldings, and the infoldings are called cristae, and here some elementary particles, stalked particles are there, and which are called oxysomes, and nothing but F0, F1 particles. So electron transport system and oxidative phosphorylation occur through various electron carrier complexes as well as coupling factor located in the inner membrane of mitochondria, including the infoldings, that is cristae, and in association with perimitochondrial space. So the different areas involved in electron transport system and oxidative phosphorylation here we represented. Of course NADH2, FADH2 are there in the matrix, they undergo oxidation there in the matrix, but that electrons they are captured and they are carried through various electron carrier complexes which are located in mitochondrial inner membrane. Here I represented mitochondrial inner membrane. This is inner membrane, lipoproteinaceous membrane, of course impermeable to protons we know, and now the carrier complexes, electron carrier complexes, complex 1, complex 2, complex 3, complex 4 with various electron carriers, and they are located in inner membrane of mitochondria, that is cristae, and in addition mobile electron carriers like ubiquinone and cytochrome C are also involved in electron transportation, which are located in that inner membrane of mitochondria. Yes. And then this is the space between outer membrane and inner membrane, as I have shown there, that is perimitochondrial space. Okay, so matrix here, perimitochondrial space, this is the inner membrane of mitochondria including cristae, and these are the complexes that are involved in electron transport system with various electron carriers including the mobile carriers ubiquinone and cytochrome C, and this is the coupling factor with F0 and F1 parts, F0, F1 particle coupling factor, the smallest rotating machine in the universe involved in the synthesis of ATP. Okay, that is a representation of the picture and mean where electron transport and oxidative phosphorylation takes place. Now, and in this scheme, I am going to explain oxidation of NADH2, then what happens and how that energy is utilized for ATP synthesis. Now let us see, and here oxidation of NADH2 I represent, NADH2 added to matrix now, they undergo oxidation, and oxidation of each NADH2 molecule in presence of the enzyme NADH dehydrogenase, and this enzyme is a part of complex 1. So the complex one itself named as NADH dehydrogenase complex with electron carriers as well as the enzyme NADH dehydrogenase, and that enzyme oxidize this NADH2, and NADH2 on oxidation liberate electrons and protons, and the electrons through this electron carrier complex received by ubiquinone, and from ubiquinone to complex 3 directly and never taken by or never touches complex 2, and from complex 3 to complex 4 through cytochrome C, and this is the mobile carrier, and then to complex 4, and finally from complex 4 that electrons are received by half molecule of oxygen. And this is the terminal acceptor of electrons. Oxygen is the terminal acceptor of electrons in electron transport system. Okay, that is the actual role played by oxygen in aerobic respiration. Of course, oxygen is essential for aerobic respiration for link reaction and Krebs cycle we followed, but oxygen do not play any direct role there. The direct role of oxygen in aerobic respiration is observed during electron transport system. Whatever that electrons liberated by the oxidation of NADH2 and FADH2, they are received by oxygen finally. So we consider oxygen as a terminal electron acceptor or ultimate or final electron acceptor in aerobic respiration, and play a direct role as electron acceptor in electron transport system and reduce it to water molecule. Here oxygen, by the time it receives 2 electrons, it take protons from the matrix which are always available in the matrix and reduce it to water molecule. Okay? And as oxidation of each coenzyme result, the liberation of 2 electrons which are received by half oxygen molecule in presence of 2 protons and reduced to 1 water molecule. So for the oxidation of each coenzyme, 1 water molecule is formed as metabolic water, that is by the utilization of half oxygen molecule, that is 1 oxygen atom, right? Now coming to the energetics, and here the electrons, they are carried through various electron carriers from complex 1 to ubiquinone and from ubiquinone to complex 3, and then from complex 3 to complex 4. In these steps, total 3 steps, and the protons from matrix, they are translocated into the perimitochondrial space, because here ubiquinone not only electron carrier and also the proton transporter, so it pull the protons from mitochondrial matrix. For 2 electrons, 2 protons pulled by ubiquinone and are added to perimitochondrial space. In the same way, complex 3 and complex 4 are also not only electron carriers, they are proton carriers also. So they also pull protons from the matrix, and every time for 2 electrons, 2 protons are pulled by these electron carrier complexes including ubiquinone. And finally, if one oxidation, that is oxidation of NADH2 results, a pair of electrons involved in electron transport system finally received by oxygen molecule, half molecule of oxygen, by the time in 3 different steps, protons from the matrix translocated and added to perimitochondrial space. Okay, already we represented here, and 2 protons here, 2 protons here, 2 protons here. So totally 6 protons from the matrix translocated into perimitochondrial space by the time that pair of electrons received by half oxygen molecule and reduced to water. So, and gradually proton concentration increases in perimitochondrial space, and gradually proton concentration decreases in the matrix. So between matrix and perimitochondrial space, a proton concentration gradients develops. According to Peter Mischel chemiasmatic theory, that proton concentration gradients, and it allows the movement of protons from perimitochondrial space to matrix again due to proton motive force. But that mitochondrial membrane is impermeable to protons, so protons directly may not diffuse through membrane. So always such protons, they are channelized through coupling factor, and here the role of coupling factor, now let us follow. Coupling factor with 2 parts F0 and F1, F0 is the proton channel that allow channeling of protons. As the membrane is impermeable, only coupling factor should channelize the protons from perimitochondrial space to matrix. And here, and F1 is the head part of coupling factor, the catalytic side with the enzyme ATP synthase. Okay, and by the time the protons, they cross the head region and added to the matrix, and certain amount of energy liberated in the head part, that is F1 part of coupling factor, and captured by ATP synthase, and that is utilized for the coupling of ADP and pi to form ATP. So, oxidation of coenzymes like NADH2 results electron transport and proton translocation, and the protons channeled through coupling factor cause the synthesis of ATP, and that is electron transport system and oxidative phosphorylation. Here, as we follow the numericals, for the oxidation of 1 NADH2, a pair of electrons, that is 2 electrons involved in ETS, finally received by half molecule of oxygen. By the time and 6 protons from the matrix added to perimitochondrial space, the same 6 protons channelized through coupling factor and again added to matrix, and the energy liberated by that 6 protons is captured by the enzyme to couple 3 molecules of ADP and inorganic phosphate to form 3 molecules of ATP. As you know that energy of 2 protons utilized for the synthesis of 1 ATP molecule, no? So, and the energy released by 6 protons in the head region, and that is utilized for the synthesis of 3 ATP molecules. So, as we summarize that oxidation of NADH2 that occur in inner membrane of mitochondria in association with perimitochondrial space, as well as that F0, F1 particle. Okay. Let us write here for the oxidation of that 1 NADH2, as it liberate a pair of electrons and cause 6 protons channeled through coupling factor and cause the synthesis of 3 ATP, and these 2 electrons with through 2 protons received by half oxygen molecule, that is the electron acceptor, and form 1 water molecule. Okay now? So, finally we can follow oxidation of 1 NADH2 there in the matrix of mitochondria results the formation of 3 ATP molecules and 1 water molecule as byproduct, that is by the involvement of half oxygen molecule, utilization of half oxygen molecule. 6 protons involved in channeling through coupling factor and cause the synthesis of 3 ATP. Students, now let us follow oxidation of FADH2. That means if one FADH2 molecule undergo oxidation, and how that energy liberated cause the synthesis of ATP and how many ATP molecules are formed. Now, coming to oxidation of FADH2, there in the matrix, it is catalyzed by the enzyme succinic dehydrogenase complex, and that is nothing but complex 2. Okay. So, oxidation of FADH2 is performed by complex 2 with the enzyme succinic dehydrogenase. So, complex 1 is not at all involved in the oxidation of FADH2, and you have to remember that one. And in the same way we can compare oxidation of NADH2 is catalyzed by NADH dehydrogenase complex, that is complex 1. There during the oxidation of NADH2 there is no part of complex 2 we followed already. So, as we compare oxidation of NADH2 and oxidation of FADH2, oxidation of NADH2 through complex 1 and oxidation of FADH2 through complex 2. Complex 1 is not involved in the oxidation of FADH2 and complex 2 is not at all involved in the oxidation of NADH2. Okay. Now, and let us focus on the oxidation of FADH2 catalyzed by succinic dehydrogenase enzyme, which is the enzyme present in complex 2, and the electrons directly received by ubiquinone, and there is no proton shifting here because complex 2 is involved only in electron transport but not in proton shifting, and it is not the proton carrier. Now from ubiquinone that electrons are received by complex 3, from complex 3 to complex 4 through cytochrome C. As usual, just like previously we followed here, that shifting of electrons from ubiquinone to complex 3 and complex 3 to complex 4 results translocation of protons also from matrix through the complexes 3 and 4 into inner membrane, that is intermembrane space, perimitochondrial space. So as a result, finally the electrons of FADH2 received by oxygen molecule, that is half oxygen molecule, and reduced to one water molecule. By the time that electron shifting results, the translocation of protons from matrix into perimitochondrial space, particularly in 2 steps we followed. So totally 4 protons from matrix added to perimitochondrial space due to the oxidation of one FADH2 molecule. Okay. And as that 4 protons are channeled through coupling factor and again added to matrix and liberate certain amount of energy which can couple 2 ADP and 2 inorganic phosphates to form 2 ATP. So as we summarize that oxidation of FADH2, of course through complex 2 and ubiquinone 3 and 4, and complex 1 is not at all involved, and finally that 2 electrons received by half oxygen molecule and form one water molecule as byproduct, but that electron transport of 2 electrons cause 4 protons channeled through coupling factor and cause the synthesis of 2 ATP molecules. Okay, no? So the energetics 2 ATP for oxidation of 1 FADH2 we consider, and as usual 1 metabolic water is the byproduct and half oxygen molecule is involved and 4 protons are channeled through coupling factor.